Okay, hi everyone. Well, today I'll be talking about some joint work with my advisor, Tamara Broderick, on learning interactions in high dimensional data. To kind of motivate this problem of learning interactions, let's look at a concrete example. So suppose a doctor has access to the electronic health records of n different patients, and suppose those records contain genetic information such as the DNA sequence of each of her patients or gene expression levels, as well as lifestyle choices such as how much sugar a patient consumes or how active a patient is. And she might be interested in a particular health outcome, say blood sugar levels, and she might want to understand, well, how do genetic and environmental factors relate to blood sugar levels? But an immediate challenge is that in general, there are thousands to millions of these covariates. So manually scanning through them and relating them to blood sugar levels is just going to be too time consuming. So instead, she might want to find a small subset of important covariates automatically and then follow up on this subset to see which actually have biological meaning. So one way to do this would be to fit an additive linear model. So maybe she runs the lasso on all of this data and finds out, hey, there seems to be a diabetes gene and high sugar diet that's moderately correlated with blood sugar levels. Well, is that enough? Well, maybe if someone both has this bad diabetes gene and consumes a lot of sugar, they could be super at risk for developing high blood sugar levels. And in general, there could be some really complicated nonlinear structure of the problem where, for example, blood sugar levels could depend sinusoidally with the time of the day. So kind of when you model interactions, the big issue is the number of interactions explodes with covariate dimension. If we have a million covariates, just looking at even pairwise interactions, you suddenly have a trillion total effects. So at least for methods that jointly model interactions, they can't scale well because they explicitly generate all interactions. And for methods that do scale better, they turn to greedy approximations. So I'll show that we don't need to turn to approximations and can actually model interactions jointly, but do so in time linear and covariate dimension by exploiting some special interaction structure. So the outline of this talk is as follows. I'll try to further motivate looking at interactions and try to highlight what's really challenging about this problem. And then I'll show how we actually go about modeling and learning nonlinear interactions. And I'll focus on setting up the problem and leave the technical details to our paper. And finally, I'll conclude with an empirical comparison with other interaction discovery methods. So returning to the example we had before of blood sugar levels, we might expect that blood sugar levels depend non-linearly on sugar consumption, where as a patient consumes more and more sugar, suddenly blood sugar levels increase more and more at a non-linear rate. We call such an effect an additive or marginal effect because it summarizes the average effect sugar consumption has on blood sugar levels. Well, now suppose a patient has high blood sugar levels, then the doctor might be able to prescribe a particular drug medication such that as the patient takes more and more of this drug, blood sugar levels decrease. But maybe this drug is only effective for patients with a particular gene A. And even worse, some patient with another gene B, this drug is actually harmful, where as the patient consumes more of this drug, blood sugar levels increase. Well, we call such an effect a two-way interaction effect because it summarizes how the effect of the drug depends on the genetic context of the patient. So in general, what we've been interested in is trying to identify out of the potentially thousands to millions of covariates, which ones drive the response and estimate these nonlinear effects. But the challenge is best understood through a simple example where suppose we had n equals 1,000 total covariates or data points and a million total covariates, and let Q be the highest order interaction. And this size problem is fairly common in genomics, for example. Well, if we just have an additive model and we look at the space of one-way effects, then storing that 1,000 by million matrix of effects is going to require about 10 gigabytes of storage. So this is something the lasso can readily handle on your laptop. But if you just bump up Q by one and look at all pairwise interactions, then suddenly you have a trillion effects, and storing that 1,000 by trillion matrix is going to require 10 million gigabytes of storage. So this is not something you can easily do. So that's kind of highlights the big statistical and computational challenges because you already started with this problem where the number of covariates was much larger than the number of data points. And then this problem just got way harder because of this combinatorial explosion of interactions and the presence of nonlinear effects. So at least on the statistical side, previous work addresses those challenges through a sparsity condition, which basically says that a lot of these effects are actually zero. But nevertheless, on the computation side, they're still slow. They scale as OP to the Q because they explicitly generate all interactions. So if Q is larger than 1 and P is moderately large, then these methods don't scale well. So I'll show how we can actually make these methods scale, make a method that scales linearly in both P and Q by exploiting some special interaction structure. So how are we actually going to model nonlinear interactions? Well, let's generalize the problem we had before and call each of our covariate dimensions from 1 to P, and let the superscript denote data point n and the response y. And we'll start with the fairly general model where the response is equal to some potentially nonlinear function of the covariates f plus additive noise, where this unknown regression function f we would like to learn belongs to this class of functions h. So to estimate f, we're going to look at a penalized least squares estimator, which is thus the minimizing function over this class of functions h of the sum of squared errors plus this model complexity term jf. So there are 4 immediate subproblems that result from this formulation. And the first is, well, how do you even pick H to model interactions? And how do you pick a penalty JF to induce sparsity? And even if you could make such choices, how do you tractably solve for F hat and report the effects in F hat, since that's at the end of the day our goal? So for the first problem, the way we kind of address this is we just look at all interaction effects of order up to Q. And mathematically, that says that we're going to look at all functions f that can be written as a sum of lower order effects fv, where fv depends on at most q covariates. And we already saw an example of this before, where v was this two-way interaction between drug and gene. But an immediate issue with this formulation is identifiability. You could add and subtract a non-zero constant to any of these components and still have the same expansion f. So we're going to address that issue through what's called the Functional Anova, which says that higher order interaction effects represent all variation that can't be explained by lower order effects. And mathematically, what that says is it's going to say that a higher order interaction effect, FB, is orthogonal to all lower order interaction effects, FA, where this notion of orthogonality is in an L2 sense, so it corresponds to a variance decomposition. So if you're at all familiar with ANOVA decompositions of contingency tables, this is the direct analog and extension, but now to functions that lie in a Hilbert space. For the second problem, it turns out to be a lot easier to induce sparsity when you think of JF as a negative log prior. So what we do is we build on existing Bayesian sparsity priors to get sparsity in our problem. And a lot of this is driven by a lot of previous work shown here. But our big contribution is really on the computation side, where we basically show how to solve this penalized least squares problem in time linear in covariate dimension and still report effects still linear in both covariate dimension and interaction order. When we look at these special types of kernels, we define as SKIM-FA kernels. But let's see actually how SKIM-FA kernels actually perform in practice. Well, in terms of comparison, what we did was we looked at methods that could scale up to at least thousands of covariates, because the problems we are interested in are pretty high dimensional. In terms of evaluation criteria, again, we're interested in trying to identify what covariates the response actually depends on and estimate those effects. But this is challenging because unlike standard machine learning applications where you can immediately turn to real data and look at out-of-sample predictive performance to benchmark methods, here real data doesn't contain ground truth. So to actually do benchmarking in real data, we construct this sort of synthetic ground truth to then benchmark methods. But what we can always do is we can always look at synthetic data first and have ground truth to compare methods. So what we do is we generate a lot of synthetic data sets, and we found better variable selection performance than these previous methods. But to kind of give you a flavor of one area where our method really shines is a setting where there's really strong pairwise interactions and weaker additive effects. So we consider a particular example where there are a total of 1,000 covariates, where 5 of the covariates actually influence the response in either additive effects or interaction effects. And the remaining 995 covariates have no influence to the response. And this is a pretty high dimensional problem because we only have n equals 1,000 total data points and 500,000 possible interactions. So in terms of variable selection performance, what we do is in green, we look at how many of the covariates that actually drive the response in either main effects or interaction effects does a method pick up. And here, the maximum is 5 and higher is better. And in red, we look at how many covariates that don't have any influence on the response, how many of these covariates does a method pick up. So lower is better here, and the max you can have is 995. So the first two methods we compare against are these two lasso methods, which assume linear interaction effects, and they jointly model all interactions, so they scale quadratically with covariate dimension. But in practice, the code is optimized in C++, so they can be applied to thousands of covariates. So basically, the way pairs lasso works is it just generates all linear interactions and then applies lasso to it. And then hierarchical lasso does a more sophisticated version, taking into account some more of the interaction structure. So in terms of actual results, all methods pick up all 5 correct covariates, but both pairs lasso and hierarchical lasso pick up a lot more fake covariates. In terms of runtime, we're a bit slower than hierarchical lasso, but eventually we showed that we're faster than hierarchical lasso because our method scales linearly with covariate dimension, and we're faster in this example than pairs lasso. The next method we compare against is SPAM, which is sparse additive model, which assumes additive effect, nonlinear additive effects, but no interactions. And since this particular synthetic data set here has a lot of the signal locked away in the interactions, SPAM does poorly on this example, where it only picks up one of the correct covariates and picks up 41 fake covariates. But in terms of runtime, it's fast because it's linear in covariate dimension, but this comes at the expense of statistical performance. And finally, the last method we compare against is Mars, which greedily traverses a space of interactions. And this method picks up most of the correct covariates, but it comes again at the expense of picking up a fair number of fake covariates. And in terms of runtime, it's similar to our method. So for real data, the way we thought of the evaluation is we took a really low dimensional data set, this bike sharing data set with 4 continuous covariates. And then we made it high dimensional by adding 1,000 covariates that have no influence to the response. So this is now kind of creating the synthetic control where if a method picks up one of these 1,000 covariates that have no influence to the response, we know that's bad. We can use that to benchmark the different methods. So in this table here, the blue represents how many of the original 4 covariates were selected. Since we don't have ground truth effects, higher is not necessarily better, but maybe we want something non-zero so that a method's picking up something at least. But the more important number is this red, where it says that a method picked up one of these fake covariates. And here lower is better, and the max you can have is 1,000. So when we do this comparison here, our method actually doesn't pick up any of the fake covariates, whereas the other methods pick up a lot, or at least some fake covariates. And in terms of runtime, it's similar to these other methods shown here in the synthetic data example. So overall, I've shown how we can learn interactions tractably through SKIM-FA kernels. And currently what we've been doing is trying to apply our methods to actual biological data sets in genomics. And if you're interested in learning more, this first paper, which I described, should be on the archive soon before the conference. And this follows up on this other work where we looked at linear interaction effects and making it scale linearly in covariate dimension, which is shown before. Thank you.